Don't ignore what's happening here. We're having some kind of powerful, weird alchemy, and you have to pay attention when that happens. Stuff is not to be treated lightly.
Listen, I actually just came out here to have a cigarette, okay, and relax and
sure,
do my thing.
Yeah.
So, maybe another time.
Yeah, but there may not be another time. Okay, I may never get this chance again. I mean, we may never, you know, be able to return to this moment.
Hello. Welcome everyone to today's workshop number 6 about proteins and protein folding. This is good. I think we have more students today than last time. And yeah. So first of all, I hope everybody is now free to go outside because the emergency state has ended and you've been able to catch some air and have some fresh minds and are able to take in a lot of information today, because maybe some of it is not going to be too easy, but I'll try to make it as easy as possible. As always, we're going to start with a little review, so you know where we a little bit are in the context of the whole workshop. And then we're going to go into the new topic of today. Alright, so let's see. Last time we had this, right? So last time we talked about motor proteins and motions. So after we finished talking about muscles, the myosin, we started talking about cilia. Right, as you can see in both pictures, these kind of hair-like structures that are moving back and forth rhythmically. Right, and you might remember that if there's only one attached to a cell, it's called a flagellum. Flagellum, I don't know. Okay, and this is also doing like a wave-like motion, right? So we talked about how this is due to a protein we looked at that's called dynein, which is usually also found transporting cargo vesicles throughout the cell in only one direction. Right? And we also talked about how these flagella are made up of what's called an axoneme, right? And in this axoneme you can find two types of dyneins, right? The outer dynein and the inner dynein. And in this axoneme, these dyneins are responsible for doing this whole bending motion, right? What is looking like a wave. And we talked about the mechanism that is involved by ATP hydrolysis. The purple part is moving from the light blue to the yellow part and thereby lifting and putting it in the, like basically reaching and then pushing it along the microtubule. Okay, and last time we had a little bit of trouble with the internet. That's why we moved the workshop back to 2 p.m. on Saturdays. So I talked about this actually for maybe like 5-10 minutes, but nobody was able to hear. And I was talking about how cilia are important for humans, right? And where you can find them in the human body. Oops, that's not what I wanted to do. I wanted to show you the video. Right, so this is a very short video that shows you actually part of a human, right? And the cilia that are on top of these cells. So these are epithelial cells. Again, okay. From the lungs, right? And your whole lungs are lined with those type of cells that have cilia that are moving in a wave-like fashion. Now, why is that, right? You might think that, well, you don't need hairs in your lungs, right? But important is the movement, because on top of these moving cilia, you also have a mucus layer, right? So slime, basically. So you have these cilia cells, and somewhere in between them there are slime-producing cells. And the slime basically catches bacteria and foreign particles, right? Dirt or something that you inhale, right? Because you're breathing all the time in and out, and your lung, if you have ever seen a picture, has many cavities. So it basically acts like a filter for the air that you're breathing. So things might get stuck or might be inhaled over a long time, and they might accumulate. But to prevent that, basically these hairs, they move the slime out of the lung, and then you can basically cough or swallow the slime, basically to get rid of it and to clean your lungs. So these are very, very important. There are also some diseases associated with those, like cystic fibrosis. I think is one of them, where the slime is basically too thick and the cilia cannot move it, so it accumulates in the lungs. It's a very tragic disease, but it's genetic, and so you don't have to worry about if you don't have it by now. Also, you might know that, of course, smoking is bad for you. This is also because how it affects the cilia, for instance. Smokers have a higher risk of lung cancer because the tar from the cigarettes is hindering the motility of the cilia. Okay, but that's only on the side. There are also cilia in many, many parts of the body that have all kinds of functions. So it's a very, very essential part for the human body. What you saw, for instance, on the picture before, right? Maybe you recognize that was a sperm cell, right? What I showed you before was from a sea urchin. But also, of course, human sperm cells have these type of flagella to move them forward and find the egg cell. And otherwise, there are also cilia found in organs. I was looking a little bit into details of where and what kind of organs, and one in particular is the kidney, but I was not 100% able to find out why. So it talked a lot about how there are some important receptors and also secretory complexes on the cilia that are important for correct function, and also generation of the kidney during embryogenesis. Yes, the other example, for instance, here are airway cilia, which we just talked about. I don't know exactly ependymal. Okay, kidney cilia, those here. Those that I mean. So there are some specific proteins on the surface of them that have a function in the big context of the kidney. Then there are also some which you might not think of as cilia per se, right? For instance, the olfactory cilia. So as you might know, for smelling, you have specific cells in the top of your nose. And these cells, they reach into your nose cavity, and they have receptors for sensing smell on the tip of them, right? And then the signal is translated into electrical signals, goes to the brain, and then you can actually smell things. So these also depend on cilia. Then we have the so-called kinocilia. Those are found in your ear. So, and maybe, I'm not sure if you know how the ear works, but the vibration from the ear is actually translated into vibration of liquid, right, in the cochlea, the snail type structure inside your ear. And the way we hear things is if the liquid's moving, those cilia are also moving, right? And then with the frequency they are moving, the signal is then translated into electrical signals and interpreted by our brain. And finally, we also have retinal cilia. Right here, the movement is not so important. Maybe most important is the structure, right? So this is a photoreceptor, basically. So the rods and cones that you might know from biology, that you use to see, for instance, dark and light, and also colors, they have like a cilia backbone, basically, that keeps them up like a rod, basically. And also there are surrounding cells that are able to, or that use cilia to provide nutrients to these types of cells. So you see, cilia are very, very important in your body, especially also in areas where there's contact to the outside and where basically signals want to be sent or received by cells. Right, so there's a wide variety of different cilia. Yes. Today, we want to look at flagella, but not the flagella that we talked about before, like the ones you find in sperm cells, today we're going to talk about bacterial flagella. Right? And if you look at the picture here, right, on the left, I'm not 100% sure now, but I think these are Salmonella bacteria, right? And I think also the same on the right. And you can see, well, their movement is different, right? So it's not a wave-like motion of these types of flagella that are protruding from the cell. What they're doing is like a rotating, like corkscrew type of motion. And this is actually a very, very interesting machinery that bacteria have invented to be able to move through medium. And today we're basically going to look at how that works. Okay, so for that, first a little bit introduction, how bacteria move in general. Here it's with the example, for instance, this could be, for instance, E. coli, right? If you remember the bacteria that we worked with in the laboratory. So E. coli also have flagella, not only one, but multiple. And usually for moving in a straight direction, these flagella, they have been found to rotate in basically all together in a bundle like this, and they rotate in a counterclockwise rotation to move forward. What then happens if they want to change direction, right? For instance, if they sense food or if they sense a chemical repellent, something that's dangerous, what they do is they basically invert the rotation of these flagella and they start to tumble, right, in a random direction. Maybe you can see that a little bit in some of them here. Maybe not so much. You can see, for instance, here is one tumbling, right? So basically it's reorienting itself by inverting the rotation of the flagella and thereby like turning in different direction, and then it's moving counterclockwise again, and so they move forward. Okay. So I talked to you about that the basic mechanism is different, so also the structure of this whole system is different. If you think about what we looked at last time, on the right you can see a representation of that, a eukaryotic flagellum, right? Maybe if you know, I hope you know. For instance, humans are eukaryotes, and bacteria you can call prokaryotes. So with a nucleus, without a nucleus. But here the eukaryotic flagellum is basically this long structure with a microtubulus in the middle that can move left and right. But here it's like something that is anchored in the cell membrane, and it is rotating basically at this point where it just protrudes from the outer membrane. Okay, so if you look closer to it, it's actually super interesting machinery, right? So you have what you call a propeller, right? So that's very similar to the normal flagellum that we saw before, with like an exoneme type of structure. And then you have a hook that is rigid. So basically that always has this conformation, but is able to rotate and like anyways. And then you have many different parts that are called very similar to something that you might have heard before, right? So you have, for instance, a bearing, a stator, and a rotator, right? So we're going to talk about the functions just in a second. first I wanted to talk to you about, so this movement that we've talked about in the last workshop and that we're going to talk about today, the main thing that is different is not only the structure, but also where the energy comes from, right? And I wanted to make that a little bit clear. And I hope you can a little bit understand the process of energy production by looking at this. So mainly in cells, energy comes from glucose, right? For instance, in human cells. In bacteria, it can come from many types of metabolism, but let's just say we talk about bacteria that also use glucose, right? For instance, E. coli can also use glucose. Something at the very center is the citric acid cycle. Probably most of you have not learned about that yet, but you will in the future. And the result of the citric acid cycle is ATP. Right? So we talked about ATP, how basically the use of ATP and splitting it into ADP and P can lead to, for instance, the movement of kinesin and denine, and also myosin, right? Something else that is happening, right, when glucose is used in the cell through metabolism is that not only ATP is produced, but protons are also basically pushed outside the cell in this intermembrane space, right? And this effect has a certain name, and well, you can abbreviate it as PMF, or it's called proton motor force, right? So maybe you can imagine it. So when glucose is used through a bunch of different proteins, protons are transported from inside the cell to the intermembrane space, right? So the concentration of protons increases steadily in this space, right? And here the concentration is low. What that does is it creates a potential energy. And maybe you've heard about potential energy from physics. Basically, it's a difference that is, or energy that is put into, well, actually, let me explain it with a simple example of, for instance, looking about how this works with water, right? We use that very efficiently, for instance, when we talk about renewable energy sources. For instance, we know that wind energy and solar energy are very, very useful energy sources, but they have a main problem. One of the problems is that they only produce energy under certain circumstances, like for instance the sun is shining or wind is blowing. So maybe they produce too much energy at a time we don't need it, and too little when we do need it. So as a kind of buffering system, this setup basically saves this produced energy, the surplus energy, and saves it as potential energy. Right? This potential energy means just that what it does is it pumps water from sea level to a higher reservoir. Right? So it uses this energy to use a pump that then increases the volume of water in the reservoir. So then the water's up here, it has basically potential energy, and whenever, for instance, the sun is not shining or the wind is not blowing, you can open this again, you shut off the pump and just let it flow through, and or you use it as a generator, right? So when the water flows back, it basically rotates the generator in the opposite direction, and it can turn the potential energy into electrical energy again, right? Okay, and a very similar mechanism happens in cells, right? So these protons have basically you can think of it as being pumped outside as being pumped, or being pumped in against a high concentration is like putting them up in a reservoir. So what they want to do actually is they want to go back. So they have a lot of energy saved up that can be released by putting or letting them back inside the cell. Okay. So let's see what it looks like in a bacterium, right? So something that happens, we just talked about the respiratory chain, or how this is actually called the respiratory chain because it also uses oxygen. And well, it's part of how glucose is used in a bacterium. So, and as a result, protons are pumped outside. And there are also some active processes that can do that. Right? So, and then you have basically a lot of protons outside, and the way they can come back is through this ring. So basically this is the turbine that I showed you in the picture before when it comes to pumping water, right? So here on the left you can see this intermembrane space, which is full of hydrogens, and those basically go back through this kind of turbine and thereby twist it or turn it, and turning the whole flagellum in itself. So this is a super smart mechanism which has evolved in bacteria, and it's also a super huge complex. So actually there has been a lot of research until now about this protein, or this whole complex. Right, so I told you a little bit like about the exonym. The hook is rigid, and then there are parts that are in the outer membrane locked, and there are things that are in the inner membrane, and there's something called a stator and a rotor. Right, so basically the stator holds the whole thing tight in one place in the membrane, and as you can see on the left, the rotor is the part that's turning. That is known. That has been known for quite a long time. And how it actually works in detail has not been 100% shown yet, right? There are a lot of theories, right? For instance, one of the theories, well, we know what is responsible for letting out the protons or the hydrogens from the intermembrane space. So we know these protons, they move back inside the cell through parts of this protein complex, which are called mod B and mod A. Right? Probably mod stands for motility or motor, something like that. So basically by moving through here and through this torque generating surface, they are responsible for turning this C ring, which consists of also other parts. And just before I talked about how also this rotation can be reversed, right? Also, this is not completely known yet. There are some theories that, for instance, the Fleegee, I think that's what it's called, is involved in that, right? By changing conformation, kind of makes the whole machinery run in the opposite direction. Okay, so going back to the example of the turbine, this is actually how it also works, or how humans use it in real life. If you've had physics for a while, you've probably seen one of those, right? It's just a simple electrical motor. So on the outside, you have basically magnets, which are surrounded by a wire. No, wait. Maybe, no, okay, no. You have wire around a metallic block, basically, and they're in a circle. and in the middle you have the magnets. Right? And they create a magnetic field, and through that the motor then turns. That's how it works. Right? And this mechanism is very, very similar to what is happening in real life with Flagellar motors. Yes, so basically, like I said, you have this reservoir of protons, and by letting them move through this tube, you use their potential energy to make something turn. Right? In this case, the output is electricity. In case of bacteria, the output is movement, right, of the whole cell, which is actually very great. So I think the speed that they can reach is about 50 times their size per second, right? That doesn't sound very, very fast. I think when I read, I think it takes him like 2,500 hours for a mile. So none of them live that long. But if you think about, for instance, a cheetah, the fastest land animal, can, for instance, only run at a speed of 25 times its size per second. Right? So half of it. So let's see. Yes, so as I told you, we don't exactly know how this whole rotation works. Right? I mean, we know it, that one thing's turning, the other one's not, and we know that the protons go through it, but the exact mechanism inside the protein have not been found yet, because it's very, very difficult to crystallize and get the models for these type of large proteins. So, but what we can look at, wait, that's not it. I will show you in a second. And here's one of the theories that I wanted to show you, and it's also a good summary of what I just talked to you about. And after this, we're going to talk about a protein that can be used as kind of an example of how this whole process could work. And it's also a very, very interesting protein, of course, and very important for cells. But first, let's watch the video very quickly. Nope.
Themselves through their environment by spinning helical motorized flagella. Rhodobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell's surface, make up the three parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called Mote A and Mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in Mote B functions in proton conductance. Each stator contains two Mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the Mote B proteins. This binding causes a conformational change in Mote A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm, The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined.
Okay. All right. So I don't think we have to look at flagellar assembly, but I hope with this, so like kind of like pistons going up and down and moving over a rigid surface, is a very interesting mechanism if you think about how to turn the rotor part. So, but again, it's not 100% sure this is how it actually functions. 1 second. Okay. So, but what we can do is look at a different protein, and one that has been easier to analyze and is not that huge. So what I want you to do is similar from the last time. So for those of you who use PyMol, let's switch to PyMol. Let's see. And for those of you who don't use PyMol, just sit and watch what I'm doing. So let's see. So from the presentation, we want to load the protein called 3J9T. Okay, 3J9T. So all you have to do is type in this command line up here, right? So you type fetch 3J9, that's 9T. Okay, there we have it. Okay, I will give you some time to do the same. Again, if you have any questions, please ask. Also get ready to perhaps draw something, because I would like for you to again speculate about the structure, right, and the function of the protein. Okay, so let's look a little bit by rotating it around. So this is also a relatively large protein. And it's probably easier to look at it if you give it some color, right? It's instead of only green. So what we're going to do is we're going to change the color and rainbow. So color is you go to the protein on the right, click on C, and then spectrum rainbow. Okay, well that looks a lot better. Right, so. And again, let's see. So let's go to the whiteboard. Okay, on the whiteboard, like always, please try to write your name. In any color that you wish. Okay, very good. Someone else? Ashton, Kate are there. Joshua? Maybe? Oh, I see. Very good. Nice. So very similar to last time, I would like you to analyze and to investigate the structure of the protein, right? So if I go back to PyMol, I'd like you to look at the structure and just like a cartoon, like an illustration, simplify it, right? Try to find out which of these, or of this protein, what parts are separate units that might have a different function than the rest, right? So turn it around a little bit, look at it from the bottom, maybe from the top, left, right, turn it around. Play with it a little bit, all right? So try to find some angles that are interesting, might give you some different insight into the function. Then start drawing your ideas. So for you, those of you who don't use PyMol, I'm going to leave it here, and I'm going to just watch what's happening on the whiteboard. Let's see. Oh, very good. Okay, what about Asta and Joshua? What do you think? No idea? Just give it a try. Kate had a good idea, right? So you definitely have a head part. Sorry, I was playing around with my... See. So just try to draw the protein on the whiteboard in basically as an illustration in a simplified way, similar what you might see what Kate did already. Of course, don't let yourself be influenced too much. Very good. Very nice. I like it. Okay, multiple colors, that's nice. So this time I would like also for you to imagine how this works, right? What this protein does. And in order to be able to do this, I'm going to give you a little bit help. I'm going to draw for you where the membrane is, right? So, you know, a membrane is a double layer barrier, right, between inside and outside. Okay. Who copied that? Okay, so I'm just going to draw the lines of where you would find the membrane. Very good. Okay, and I'm also going to give you a tip, right? Because before I talked about how this protein is very similar to the flagellum, right? So it's also basically driven by the proton motor force, right? So it basically works with a flow of hydrogens through it, right? So I'm not going to exactly draw it for you how it works. So there's a gradient of hydrogens, and then something happens, right? So what do you think? How does it work? What I want you to do for that is maybe choose a part. Very good. Choose a part and try to guess a function for it. Right? For instance, you can say, okay, oh, you can draw a line to it, and this is, like, for instance, fixed. Right? Or you can say another part is moving. And this is a very good question. So, does it spin? Answer is yes, it does spin. So, but what do you think? What spins in this case? See, so that everybody can see what we're drawing. Okay, what do you think? What spins? The shooting area. Not quite, not quite. Okay. Very interesting idea. So it could be that the outer part spins and the inner part is fixed in the membrane. All right. Any other ideas? Oh, good. Very good. Kate says, or maybe the base spins, right? Also a very good idea. But of course, to get the answer, you would have to take a look at the actual protein, right? And yeah, very good. So they are either way would be possible or would be imaginable looking at the model. And let's go and look what is actually happening. Okay, so let's take a better look. So similar to the other protein, the flagellar motor, which we talked about, in this case, in the flagellar motor, the stator was on the outside, right? In this case, it's the same. So the fixed part are basically the orange and the yellow parts of this protein, right? Those don't move. They don't move, and what they also do is they hold this head part in place. Right? So both yellow and orange are anchored into the membrane, and they hold the whole like light green and light blue part in place, so it does not move. Therefore, what's left is the rotating part, right? So stator rotor here, the rotor is also in the center and is turning, right? Because it also, this kind of looks like a wheel. Or is, if you would imagine the center of a turbine would also look like this. And importantly, what connects this turning part to the top part is this long alpha helix in the middle, the dark blue, basically. So I can also already give you the tip. So in the bottom, this wheel type thing, the rotor part, is what creates the rotational energy, right? Things just don't rotate by themselves. You have this proton flow through the membrane, which goes through this wheel and thereby spins it, right? This spinning, or this energy, is then transferred through this dark blue like line, which pierces through the middle of this head part, and then basically turns in the middle of it, right? So if you, maybe it looks a little bit convoluted, but there are basically two alpha helices in the middle of this head part. This head part is not one single part, it has 6 different smaller units, right? So each has a different color here. So light blue, green, a little bit more light blue, greenish, and so on, this gradient, right? Those are 6 different parts. And what it does, basically, the blue part is a little bit rigid at the top, and during the turning, it pushes apart different segments of this head section. Well, actually the other way around. But we'll get to that in a second. Because this protein, or this type of function, can work in either direction. So either basically the protons flow from top to bottom and thereby turning it, or the wheel is being turned at the top and thereby moving the protons in a different direction, in the opposite direction. Okay, very good. So far to what we can see with the structure and the function. For us, this is all we can do with PyMol at the moment. For more, you would need the same protein in different conformations and in different, basically, screenshots that you can lay over each other, like we did with the myosin, actually. We had this moving animation of the myosin tail, which it was swinging as like the power stroke of moving a muscle. But in this case, actually, I tried it, and the file is a little bit too big, and it always crashed my PyMol. So I'm going to rely on publications and just figures that they show from the different states. So unfortunately, we cannot see the animation today. So if you go back to the presentation, I want to show you a little bit more in detail how this whole process works. So if you were to very in detail separate all different units of this big protein complex, you would have all these different subunits, right? So you have like A, B, which are repeating. There are 3 A's and 3 B's. You have A, B, C, D, E, F, G, H, and then also, the lowercase A, B, C, D, E, right? This one is actually a little bit different. it's a protein which functions almost identically. And also for you to know, this is called an ATPase, right? So ATP, everything in proteins, something that you should remember, everything that ends in an ase is basically splitting something, right? In this case, it's splitting ATP and uses its energy. Right. So you have this top part, the different segments. You have the stator, which fixes it into the membrane that the top part doesn't move, and then you have the rotating part at the bottom. Okay, so now before we go into the mechanism, I wanted to also mention a little bit about how these structures come to be, right? So I mentioned before that the flagellar motor is actually not well understood yet because it's very difficult to get a structure, right? To actually get the data and then 3D model it to then analyze it. Part of that, or just a little part, I don't want to get into the details of how you get a 3D model yet. Maybe we'll go into detail in the future. But I just want to talk about one aspect, and that is the resolution, right? So when you do all the things, if you get a protein into a crystal shape and then you do like X-ray crystallography, what's called, you then afterwards you can do some mathematical transformations, and you get a 3D model, right? But that's actually not exactly true, because you don't get a model from that. What you get is an electron density map, right? So an electron density map shows you basically as a mesh, as you can see in the pictures on the right. I mean, the raw data looks something like this, right? So you have areas where you have high concentrations and low concentrations. You have like valleys and mountains. And from that, you can then calculate a mesh, which you can see in 3D. What then happens is you can put different structures for amino acids into these meshes and build the protein from that. I think in the future we might be able to do that, and I hope I can figure out how to teach you how to build a protein from the electron density map. But for now, I just want to talk about that this electron density map can have very differing quality. It depends on the method by which the electron density map is produced, and it also depends a lot on the quality of the protein. So there can be a lot of differences in resolution, and resolution hereby means how well you can see structures from the electron density map. Here you have A, B, C, D, and all four different pictures are the same molecule at different electron density map resolutions, right? Logically, the lowest one being the one at the bottom. With this one, you just have a blob, basically. So from the blob, it's probably very difficult to tell which actual amino acid goes into this place. With C, it gets a little bit clearer. B, you can actually see the hole in the actually I forgot the name in English. Well, I would just call it a benzoid ring. And here you can, and at the best resolution, you can actually make out different atoms, which is kind of, I would say most protein models, they are somewhat between B and C in resolution. C are the worst ones and B are the better ones. And a lot of this also relies on the size of the protein. So if you have a very large protein, it's not only hard to crystallize, also the the actual electron density map will not be as refined as you want it to have. So it will be difficult to model. And especially when it comes to detecting how proteins wander through it. Protons are very, very small particles. So probably in B and C, they wouldn't even show up. Oh, actually, they wouldn't show up. But so just recently, what I talked about, these kind of structures could be analyzed by function because they were known, and there was a little bit also known how they work, but actually proving how they function was a completely different thing, right? They had a lot of ideas how they could work, but then actually getting the picture and getting the 3D model and showing how they work took a lot longer than actually the discovery. So for instance, for this one was only, I think, 2 or 3 years ago that the technology was as far to actually make a model that was basically detailed enough to show the actual function. And for this, I would like you to also load these two models into PyMol. Right? So one is called 2WIE. Actually, I have to write that down really quick. And the other one is 3U2Y. Okay. So if you're using PyMol, just go to PyMol, use the command line. Right? And then again type in fetch. And then 2WIE. Okay. And then also type again fetch and 3U2Y. Okay. So this is going to move a little bit weird because you still have this big protein. Like basically you have 3 proteins now in one file or in one window of PyMol. And so it's going to be a little bit weird navigating it, but I believe you can do it. Remember, you can also move around with middle mouse button, and you can always center on the protein that you want by just choosing it in the right bar, then clicking on action or A, and then go center, right? So if you, for instance, want to go back to the big one, then just choose that and go center, and then you have it here again, and you can rotate it as a center. But we're interested now in these other 2 that we just downloaded. So these are part of the big molecule in the background, right? So what do you think? What part is this? What part of this protein is it? And if you look at it from different angles, particularly this angle, you might be able to see where this is from. Right? So let's try to find this part in the bigger protein. Go back. You can see it's basically one segment of this. Or basically like the dark blue part. So one segment of the rotor is what we see in the models that we just downloaded. So, and the reason we downloaded those is because I want to show you how it works, right? So I told you it uses the force of the proton gradient. Yeah, well, how does it do that, right? How can a proton gradient be turned into actual physical movement? Right. And of course, the answer is because of the rotor part. It might be a better idea to show you before talking about it. Okay, so this is why I have this illustration. So it looks a little bit complicated, but it's not as complicated as you might think. Right? So actually, the change between the left and right only happens in this left part. Right? This is part of the rotor, of course. The loops that you see are part of the alpha helices that form this rotor. And you can see something that is part of the stator, something that is locked in the cell membrane and doesn't move. So this part rotates in the cell membrane and touches very, very tightly with the subunit part A. Right? So maybe if we go back, or we don't go back. Right? So we have part C and part A. And basically what we're looking at in particular is here this area. Altogether, this is called the V0 part, but that's not so important. Okay, so if we go back, okay, this part, right? So now we have a lot of protons in the intermembrane space, right? Like I told you about. Because of using glucose, all the protons are transported outside. So the concentration of them is very high. So now, here is a very important amino acid, an arginine, and another very important amino acid, glutamate. So, in this position, basically, the glutamate is able to accept a proton. Right? Right now, it's basically complex or has an basically electronegative or like a electromagnetic attraction to the arginine, which is mainly negative, and glutamate would be other way around, positive and negative. So they're basically in place. So now what happens, these other glutamates in this kind of barrel, right, these alpha helices have all the same sequence. Right? They all have a glutamate in this specific position. So now these are loaded with hydrogens, right? They are protonated. So now this one has an opening towards the inside, right? There's like a tube for the proton to go out and be released into the inside. And that's what it does by chance, right? So bonds are not 100% fixed forever. They can be released. And when that happens, basically, this glutamate releases the proton and it changes its formation into this stance, basically. What that induces is basically this arginine then flipping over in picture B. So it flips from the downward position to the upward position. What that does, it opens the channel for protons to go onto the glutamate, which was free in picture A, and then load it with, then basically put it into the loaded position. Okay. And this is where the rotation basically takes place. From this, those protons go once around the whole ring of the rotor and are then released into the inside. Okay. So if we look at PyMol, let's see. First, we need to, as I told you, we need to see the glutamates, right? So we have to display and then click on sequence. I know you cannot see it in the live stream, but in the very top row, click on display and then sequence, and then you will get this this bar with all 3 proteins in their sequence. So we are interested in only the bottom 2, and we are interested in a glutamate. So I'm sure you probably don't remember, but glutamate has the letter E. So if you go through the sequence in the top, there should only be very few E's. Actually, for the pink one, I think there's only one here. And then go to the next alpha helix and also select the glutamate here. And in the next alpha helix as well. This one as well. And this one as well. Okay. So you can see them selected in PyMol with the pink dots, right? And also in the light blue structure. But here there are actually some other glutamates. The one I'm talking about is under 61. Alright, so select this one, select the next one as well. Select this one as well. That one as well. Okay, so we have all of them selected now. So now we can basically make them more visible by going on the right side to selection, right? And first we can give them a different name. We just call them glutamates. Right? Giving a different name you can do by clicking on A and then rename selection. And then just type in the name that you want to give it. And then it says glutamates, right? So then we want to show them not as a cartoon, but we want to show them as licorice. Right? So now you can see, basically, they are sticking out of the cartoon. Next, to make them more visible than they are now, we want to give them a different color. So we go to, this time we're going to give them a color by element. Right? And color by element, we're going to choose, what is that, black? No, okay. We're going to choose color by element and maybe yellow. Yellow is good. A strong color. Okay, very good. And voila. What we can see is now you can actually go closer and we can look at the conformation of this glutamate. Right? So this one is in the loaded formation, right? While on the other side, this one is in the open formation, so unloaded. You have to look a little bit from the top. Unloaded. And these ones are loaded. Okay. Also something that I wanted to show you is every time you look at a protein in PyMol, you can also take a look at the electron density map. Let's take a look. So for instance, some proteins, if they're modeled and if they're built, they can have some flaws on them, right? And some people actually try to cheat a little bit by making some amino acids be in better formations that support their hypothesis. But in this case, let's check it. Okay, and so you can check it by basically going to show, and then it's called mesh. Mesh is the electron density map. Oops, wrong one. Right, and this is what it will look like. So this is actually an extremely well done electron density map. It has a very high resolution, probably I would guess around 2 angstrom. You probably don't know what that is, but we'll talk about that in the future. So, and you also see that there is somewhat like a dent in it, right? So if you look from afar, or maybe zooming a little bit closer is easier, you can see that basically next to the glutamate there's basically an open hole like this. This is basically where the loaded conformation could swing into and then be stabilized. Same goes for all the other ones, right? Okay, if we could also look at the other one, for instance. So show mesh. Oops. Yes, also extremely well refined electron density. Okay, going back to the presentation. So this whole mechanism, if you look at it more simplified, is you can compare it actually to a water wheel, right? Actually, let me show you the picture of the water wheel. No, maybe not. So I hope you know how a water wheel functions, right? Water is running into the shovels and then it turns by just the force of gravity. But this, in this case, is the proton motor force of the protons themselves. So they go into the shovel, kind of like that, which is the glutamate in this position, and then the force of them wanting to go out on the other end is turning this rotor part. And then the glutamate in this position releases it again. And the atom that swings in between, is this arginine, right? Which closes the one on one side and then opens the other and reverses by flipping, right? Opens this up for another proton to go in, and then this one is closed. Okay, very good. So this is what happens in the rotor part. Now, what happens in the head part? So we said that the rotation of the bottom part is connected to the head part. So the head part is actually responsible for the hydrolysis of ATP. Hydrolysis just means splitting it up and using the chemical energy that is produced to switch conformation, right? So, and what happens, we talked about 6 different subunits, A, B. And each A, B what do you call that, hetero complex maybe, is able to bind an ATP and hydrolyze it. So I actually don't want to go into detail too much. I just want you to kind of imagine that through binding, hydrolysis, and then release, there are different states of the subunits that are affected by the rotor in the middle. Because you can see it's not quite even, right? And by opening up, by coming closer together, right, they can actually rotate the center part. Like a wedge, basically. You can see that basically the middle rotor part is always in the position where both subunits are basically leave more space. Right? So here would be in the up position, here would be in the bottom right position. Right? Because there is space where it opens up. If ATP is bound, it's actually those subunits move closer together and become tighter, and thereby forcing this center part in the area which has more space. And the switch of this tight and open position is able to do basically a forced 360 turn of this middle loop part, and thereby turning the center. Right? Together, if you see it as a cross section, we talked about what happens at the bottom, goes in and goes out and turns. And we talked about the top, right? Here the wedge part in the middle is just indicated by a arrow. Right? So it goes in 120 degree steps and it turns from one to the other. So both can happen in both ways. That's the genius about this construction, right? So one, this whole protein complex can use the energy of the protons to make ATP, or it can use ATP to produce a proton gradient. Okay, here's the water wheel. Sorry, that's in the wrong position. I wouldn't have needed that picture before. All right. Okay, so here are both versions visible, and you can see how very similar they are. So the left one is called an V-ATPase. So because it's called an ATPase, it's the one that splits ATP, and this one would use the energy from ATP to produce this proton gradient. Right? It actively transports protons across the membrane against the higher concentration on the outside, for instance. Whereas the ATP synthase, which looks very, very similar, it just doesn't have like 3 of the stator parts. The ATP synthase is basically using the power of the hydrogens, which go from outside to the inside, and then by turning the top part to produce ATP. Here's an animation of that to make it a little bit more visually appealing. Right? So they go from the inside to the outside and use this energy to produce ATP. Right? So here's the turning part. And here those basically like clamps, they open up and then close back together. And the closing part can make ATP, or it can be opened up by hydrolysis of ATP. And basically this whole protein, this kind of genius design which was found by evolution, is very, very, very similar to what we use in hydroelectric energy, hydroelectric electricity, what I showed you from the beginning. So you can either use energy and pump the water up the hill, or the water flows down the hill through the pump and produces energy. Okay. And with this, I know it might not have been too easy today, but I hope you saw the ingenuity of this whole process, which is happening in, well, essentially all cells. And I think next time I'm going to talk a little bit more about what the actually implications of this mechanism are and why it is so important. Because I think, in a way, this protein is basically the basis of life. Without this, no life would exist on Earth. Because this is the main machinery which turns, which produces basically ATP in cells. If you think about plants, if you think about animal cells, all of them need this. It's an integral part. Okay. And with that, I'm open for questions. Right, so I'm going to give you a little bit of time, because probably the delay will be quite bad by now. But I'm just going to wait for a little bit and write your questions in chat. Or if you want also to more do it via conversation, after this I will be in Sokoko, and we can also have, like, for instance, play games like we did the last couple of times. All up to you. Okay, for those of you who are leaving, don't have time, thank you for coming, of course. Okay, so if there are no further questions, I think today we went a little bit, maybe too much into detail. I think, well, personally, this is my favorite protein. That's why I wanted to talk about it a little bit more in detail. Next times, we will talk about probably a little bit broader about proteins, about what they can do. And I hope everyone will join me at this time. Next time, probably Saturday, same time. Thanks for coming. See you then.